thank you all very much for allowing me to share some ideas with you. I'm really happy to be here, even if not in person. And because ALIFE is perhaps my favorite kind of audience. I think that the ALIFE mindset is really unique and kind of very uniquely and powerfully positioned to make advances in many areas that are important for humanity and beyond. Unfortunately, I can't be there in person today, and since I am going to be talking about a robot that helps us build biological robots, I thought it would be appropriate to try to give this talk through a robot. I hope that's working. So there's my avatar for today, and I want to talk about three things in particular. I want to describe the symmetry between what it is to be an active agent navigating the world and the way that we do science and the way that we learn about the world around us. I think these things are fundamentally very similar. And then I want to go over some of the special capabilities of the biological material. You know, in ALIFE, we are often trying to take inspiration from living matter, and there are aspects of biology that you may not have seen before because they don't feature prominently in typical discussions of advances in biology. And then towards the end, I want to introduce you to a new kind of embodied mind, a new colleague in the field of synthetic morphoengineering, a synthetic robot scientist with whom we are collaborating to hopefully make some important discoveries. So those are the three things I would like to show you today. Now, in my group, we try to do this kind of a cycle that goes back and forth between understanding life as it could be and understanding mind as it could be. We're interested in understanding how embodied minds exist in a physical universe. We try to use computational tools to understand what the biology is doing at a fundamental level, sort of abstracted from the contingencies of its current implementation. And of course, we're very interested in what the study of life has to tell us about the kinds of minds that can be made by computational and other means. And so in my group, we use AI and other tools to try to communicate with all of the layers of the biological system, so all the way up from molecular networks to cells, tissues, organs, and of course the organism itself. And we have several goals. We spend a lot of time trying to build tools to enable us to detect, communicate with, and to have ethical interactions with a very wide range of diverse intelligences that are, I believe, already exist all around us, but certainly there will be many more to come. As a model system, to try to shape and improve our intuitions and our tools, we use collectives of cells, so groups of living cells, navigating the space of anatomical possibilities. We have other models too, but this is a prime workhorse to try to understand a very unconventional mind that works at a different scale in a different space and so on. And the applications of this kind of work range across regenerative medicine, of birth defects, of injury, cancer, aging. So we develop those kinds of things, as well as applications in synthetic morphology, and then some AI and ALIFE as well. So the first thing that I want to discuss is what I think is a very interesting aspect of how to do discovery and how to navigate one's world. There is a spectrum of cognition, all the way from simple machines with whom you can only interact by modifying the hardware, so rewiring, and then all the way up through systems that are amenable to the tools of cybernetics or control theory or behavioral science, such as training, and ultimately through psychoanalysis and psychology and love and friendship and those kinds of things. So it's a very wide spectrum. And where a given system lands on the spectrum is not obvious. You can't know that from a sort of philosophical armchair. You have to do experiments and find out. And I think it's very important to realize that there has to be an impedance match between the tools you're using and what you're able to see. So, for example, I think that physics sees mechanisms instead, not minds, because generally speaking, the tools of physics are low agency tools. The interface that it uses, rulers and voltmeters and various other things, are actually pretty low agency tools. And so, of course, all they see is mechanism. If you want to study minds, you have to use high agency tools, aka other minds. It takes a mind to recognize a mind and to help you as either a scientist or a being living in the world to identify other agents. And then, of course, people like Wheeler have thought about what it means for the entire universe to be observing itself and how much how much capacity to detect agency there is in the large scale process like this. And so I want to paint a picture of both embodied life and science as a kind of active inference. So in both of these processes, what you have are predictions. You try to make observations by interacting with the outside world. Those will inevitably give rise to some kind of discrepancy around what you thought was going to happen and what actually happened. And that leads you to do two things. It leads you to change your priors, and it also leads you to change the observations that you make next. So it's a continuous cycle, and this is how we do scientific discovery, but it's also how all agents navigate the world. And the original diagram here had a brain doing this because in this paper they were studying the kind of standard idea of a brainy organism navigating their world. But I think that all of the different kinds of beings across the spectrum, so from single cells all the way up through humans, and the different modifications that we can make, all of these are in fact part of that same active inference cycle. So in biology, we engineer with a very new kind of substrate. I mean, people have been engineering passive materials like wood, metal, Legos for thousands of years. Then we had active matter and now even computational matter. But I want to talk about something very special, which are agential materials. And this is what happens when you engineer with life. So to understand to what degree your material is an agential material, meaning from an engineering perspective, to answer this question, these are the kinds of things you have to ask yourself. How much of what it does do I not need to micromanage? How much autonomy does it offer? How much is what I do basically communication as opposed to micromanagement? How much of my task is reverse engineering as opposed to engineering? That is understanding and managing what the system is already doing. Very importantly, how much more do I get out than whatever my materials or my algorithm put in? I have to understand the intrinsic motivation and the agendas of the material that I work with, and what kind of tools, all the way from rewiring to some sort of high-end psychoanalysis, are going to work best for me, right? And the living matter is actually very high on all of these kinds of metrics. And so for the next few minutes, what I want to do is show you some special properties of living matter. As a biologist, and in particular a worker in regenerative medicine, one of the things we're most interested in getting to is something I call the anatomical compiler. So someday you will be able to sit in front of a computer, you will be able to draw the animal, plant, organ, biobot, whatever. You'll be able to draw the being that you want. And what the system would do is compile that down to a set of stimuli that would have to be given to specific cells and groups of cells to get them to build exactly what you draw. And it could be anything. It could be things that have existed before. It could be completely novel things like this three-headed flatworm. This is the end game for understanding how to interact with the material of life in biomedical settings. Now, it's obvious why we need this. If we could do this, if we could tell cells what to build, we would be able to basically abolish birth defects. We would be able to regenerate after traumatic injury, reprogram cancer, defeat aging, and all of that. Now, you might wonder, why don't we have this? You know, you've been hearing about genetics for decades, but molecular biology, biochemistry have been going very strong for a long time. Why don't we have this? Because we don't have anything remotely close to this. And that's because this anatomical compiler is not something like a 3D printer. It is not for micromanaging the cells or the molecular pathways inside of them. It is a translator. It's a communications device that's designed to take the goals of the engineer or the scientist and translate them to be the goals of the goal-directed living system. That is a very unconventional view of biology. That is certainly not how most biologists think about it. And that, I think, is the barrier that's holding us back from transformative regenerative medicine. Because what all of us are made of is something like this. This is a single cell. So this happens to be a free living organism called the lacrimaria, but you get the idea. There's no brain, there's no nervous system. If you're into soft robotics, you're probably very jealous right now. This is an incredible degree of control over the system. It's handling all of its local metabolic, physiological, structural needs without a brain and anything like that. And all of us made the journey from a single cell, so fertilized oocyte, that's self-assembled into one of these remarkable morphologies, or even perhaps something like this, which will then make statements about not being a machine and being something very different than something like this, which people think of as being amenable to the laws of physics and chemistry alone. And so how did that happen? How did this the process of self-assembly and the emergence of a high-level mind from what initially looked to be just physics and chemistry? And so we are made of these kinds of things. There are agendas and competencies all the way down. But in fact, even inside of this little creature, the chemistry, okay, the molecular pathways inside are already capable of at least 6 different kinds of learning. And this is something we discovered recently, is that just the molecular networks, never mind the cell or the nucleus or anything like that, just the molecular networks alone, as a free gift from mathematics, actually, it ends up that they can do several different kinds of learning, including Pavlovian conditioning. And we are in our lab trying to take advantage of this for drug conditioning and various other medical applications. Even the material inside of cells has certain cognitive properties. It can learn from experience. And so we are made of this kind of multi-scale competency architecture. This is the crucial aspect of biology, is that it is not a single layer intelligence. It has intelligence baked in all the way down, problem-solving competencies in different kinds of spaces. Okay, and so that architecture has many implications. I only have time to show you a few, but they're quite interesting. The first is that it makes on-the-fly immediate adjustments to entirely novel scenarios. So what I'm showing you here is a tadpole of the frog Xenopus laevis. Here's the mouth, the nostrils, the brain, the gut is back here, and this is the tail here. What you'll notice is that we've prevented the 2 primary eyes from forming, but we put an eye on its tail. And when you induce an eye to form on the tail, it makes an optic nerve. That optic nerve here, you can see it. It does not go to the brain. It sometimes goes to the spinal cord, sometimes to the gut, sometimes nowhere at all. But we've made this machine that enables us to automatically test and train these animals for vision, so visual cues. And what we find out is that they can see perfectly well. Now, this is amazing. This system is a radically different sensory motor architecture, and it does not need new rounds of selection, mutation, adaptation, evolution. You don't need any of that. Out of the box, you make a completely different creature where the eye is now on the back. It doesn't connect to the brain. Who knows how the signals are actually getting up here? We don't know. But all of this works out of the box. You know, zero shot learning. It works out of the box because the material is so plastic. Because the material is primed to solve problems every single time. Evolution does not make, I think, specific solutions to specific problems. It makes problem-solving agents, in fact, multi-scale problem-solving agents. So you see the degree of plasticity here. Now, another amazing thing about the material is that it facilitates memories to move through the living matter. So this is a flatworm, a planarian. You'll hear more about this later in the talk. But there's a couple of interesting properties. One is that they're highly regenerative, so you can chop them into pieces. Every piece gives rise to a perfect little worm. The record is something like 270 pieces or something like that. And they're also smart. You can train them. So if we train them for recognizing these little laser-etched bumpy areas as safe locations to eat, so this is place conditioning. So you train them, then you cut off their head, which contains the centralized brain. You have this tail. The tail does nothing. It sort of sits there for 8 or 9 days. It has no behavior. But eventually it grows back a new brain. And when it does grow back a new brain, you find out that these animals remember the original learning. So memory not only persists the removal of the brain, the somewhat more interesting thing is that wherever it's located, it is actually able to be imprinted onto the new brain as the brain develops, because the brain is what drives the actual behavior. So the information is the learned information throughout its lifetime is interacting with the morphogenetic information. So as the brain is being built, and you have to remember what does the correct planaria head look like, you build the brain, and then you have to imprint that information onto the brain. So the results of learning are somehow moving through this tissue. But there are actually even more tricks up biology's sleeve here, which is illustrated by the following. So this is a caterpillar. Caterpillars have a particular kind of brain that are suitable for their 2-dimensional lifestyle. They crawl around, they eat leaves, so they have a soft body architecture. So they need a controller that's suitable for soft body robotics. You can't push on anything. There's no hard elements. So they have a particular kind of brain. Well, it was found that if you train caterpillars for specific tasks, that is, for example, to recognize certain color stimuli and then to go over and eat the leaves that are under that color, when they turn into butterflies, what they basically do is they completely refactor their brain. okay? Because they're going to be in a hard body flying through a three-dimensional world, completely different creature. They need a new brain. So most of the cells are killed off, most of the connections are broken, totally reconstruct the brain. And the first thing that was found is that they still remember the original information, and that's remarkable. Is how do you store? I mean, we don't have any computing devices, to my knowledge, that allow you to completely trash the memory medium and then still remember the information. But there's something deeper going on here, which is that the specific memories of the caterpillar are of zero use to the butterfly. They're completely irrelevant. It doesn't move the same way. It doesn't want the same things. It doesn't want the leaves. It wants to drink nectar from these flowers. So you can't just maintain the fidelity of the information through this process. You actually have to remap it onto a completely new body architecture. Right? So again, this notion of biological plasticity, it's not the body shape that remains. All of that is plastic and can change, just like in that tadpole. And the information is not exactly persistent in its old form. It transforms. Right? Biology is the study of the transformation of memories and of goal states and other properties through different physical media, adaptively and appropriately to their new context. We don't even have the beginnings of an understanding of how exactly these kinds of things are remapped onto new embodiments. And so one important aspect of this is that biology solves problems in many, many different kinds of spaces. There's a lot of discussion, especially in AI and things like this, about embodiment. And the idea is that, well, you have to get around the three-dimensional world. You have to engage with the world to have real intelligence. But one thing that biology teaches us is that we as humans are totally obsessed with the three-dimensional space because of our own evolutionary history and our own sensory motor architecture. We're okay at recognizing medium-sized objects moving at medium speeds, and so to solve problems in 3D space. And so birds and primates, and maybe a whale or an octopus, we can kind of recognize. But biology has been doing that loop, that sensory decision-making effector loop, in other spaces long before nerve and muscle appeared. So cells navigate a very high-dimensional space of gene expression states, of physiological states, and of anatomical morphospace. So just because you see something that doesn't look to be physically embodied, meaning it moves through the three-dimensional space, doesn't mean that it's not embodied and it isn't doing that loop that's so important for agency and for intelligence. And so within that space, I just want to show you one example of problem solving in the transcriptional space, and then we'll talk about anatomical space. So we discovered with these planaria that if you put planaria in a solution of barium, and barium makes it impossible for them to pass potassium ions, so many cells are very unhappy, especially the neurons in the head, and their heads explode. And their heads explode, but if you leave them in fresh barium, you find that after a little while, they regrow a new head, and the new head is totally adapted to barium, no problem whatsoever. And if planaria don't encounter barium in their life, or in fact in their evolutionary history, there's no pressure to deal with this crazy new stressor, and yet it's kind of Moravec's paradox. You have a system that has tens of thousands of possible genes and can up and down regulate. So a huge number of effectors. How do you know which of those to fire to solve a new problem that you haven't seen before? It's a little bit like sitting in a nuclear reactor control room. There's just an incredible amount of buttons. What you don't have time to do is to start pushing all of them to see what happens. You have to very rapidly navigate a high-dimensional space and solve a problem you've never seen before. And planaria do this. They identify less than a dozen genes that allow their heads to now grow in this completely physiologically novel environment. So this is the kind of problem solving that we need to understand, both for the biomedical side and for the robotics. They're managing a space in which you can walk in tens of thousands of directions. So now I want to talk about anatomical decision making and anatomical intelligence. I want to start by briefly introducing this creature. So this is an axolotl. So these guys are also incredibly regenerative. They can regrow their eyes, their jaws, their limbs, the spinal cords, ovaries, portions of the brain and heart. Just amazing. And the first thing you realize is that morphogenesis, or the assembly of specific bodies from cells, is not just a kind of open loop emergent process. It actually is a process of error minimization. So if they lose a limb anywhere along this axis, the cells will immediately spring into action. They will restore the structure, and then they stop. That's the most amazing part of this is they know when to stop. When do they stop? Well, when the correct structure has been completed. So what happens is that the system can tell it's been deviated from its correct location in anatomical space. They get back to within some error tolerance of error, and then they stop. Okay, so error minimization scheme. But what's really important here is that it's not just about damage. This is not about repairing damage. And I'll show you one important example, which is what happened when they took axolotl tails and grafted them to the flank of the amphibian. That tail over time remodels into a limb. And so take the perspective of these little cells up here at the very tip. They are tail tip cells sitting at the end of a tail. There's nothing wrong locally. There's no injury. There's no damage. Why are they turning into fingers? Because this system, just like our cognitive system, is very good at taking very abstract, high-level goals and transducing them down into the molecular components that are needed that need to be aligned to implement that goal. Individual cells don't know anything about what a finger is or how many fingers you're supposed to have, but the collective absolutely knows. And we know that because you can intervene and it will continue to make the right thing. And so what happens is that the collective knows that you need a limb there, not a tail. And all of that percolates to align the downstream components to give you what you want. And it must be, if you were a cell here sitting at the tip of the tail, you might have no idea why are you turning into fingers? Why this transformation? And why do all these things that keep happening seem to be aligned and coordinated in a way that doesn't seem to make any sense to you? It's a kind of, almost like a kind of synchronicity that all of this seems to be happening, but you don't have any understanding why. There's a higher level system that is making this happen. And of course, this is our everyday experience too. When you wake up in the morning, you might have very high level social goals, research goals, financial goals, career goals, whatever you have, very abstract things. In order for you to get up out of bed and actually execute on those, they have to make the biochemistry dance. They have to make ions move across your muscle membranes in order for that to happen. Each one of us is this amazing example of mind-matter interaction in voluntary motion, in that very high level goals in weird spaces that our body machinery doesn't know anything about can actually trickle down to align the internal parts to make these things happen. And I want to show you an example of how this happens. In particular, in my group, we study a lot the bioelectrical interface that groups of cells use to store goals and to communicate those goals to each other in networks. I'm going to play this video momentarily, but I want you to just look at the before and after frame. These are the colors, by the way, indicate voltage. So we have ways of measuring the voltage of any given cell, not just neurons. Every cell in your body has voltage potentials, and they all make networks that process information. And so what you'll see is that this is the before. And so this cell, which has a very different voltage than the rest of these cells, right before it gets touched, it's different, but boom, now it has been hacked. So look, so this blue cell here, minding its own business, moving along, this thing reaches out, touches it, bang, that's it. That's all it takes, is this tiny little touch to communicate a bioelectrical state that totally takes over what the cell is doing. And now it's going to come back and it's going to be part of this collective, and it's going to work on something. Learning to pass along these convincing messages is really important. And I'll just show you two very quick examples of how we use them for regenerative medicine. So here, what we do is, into this early frog embryo, into some cells that are going to make gut here, we introduce an ion channel that establishes a particular voltage state. And the voltage state just says, build an eye here. That's all it says. It's a very high level subroutine call. We don't know how to micromanage the process, thousands upon thousands of genes necessary to make an eye, to tell the stem cells what to do. We don't know how to do any of that. What we do know is how to say to the material, build an eye here. And when we do, here it is. Very nice eye built where a gut should be. We didn't have to micromanage any of it. And by the way, the material has this amazing property. If you only get a few cells, so in this cross section through a lens sitting out in the tail somewhere, the blue cells are the ones that we injected. Well, there's not enough of them to build an eye. And so what do? They recruit their neighbors. So all this other stuff out here, we never touched it. It is building an eye in cooperation with these cells because they are instructing it. So we didn't have to teach it to do that. It already knows how to do that. We say to you, build an eye, and they figured out that, hey, there's not enough of us. We need to recruit other cells, much like other collective intelligences like ants and termites will recruit nestmates to lift heavy objects and things like that, right? So this is the body is in fact a collective intelligence. It solves problems in physiological and gene expression spaces, and of course in anatomical space. And we can use this bioelectrical interface to communicate with it and ask it to do new things. This, to me, is not only an application heading towards the biomedicine of organ regeneration, but an example of learning to communicate with a novel intelligence that works in a space that is very hard for us to visualize. We've learned how to do some other things. For example, to change the memory of that collective intelligence of what the right pattern is. So here is the planarian, and what you can do is chop off the head and the tail, and this middle fragment will reliably build one head, one tail. How does it know? How does it know how many heads to build? In fact, these cells back here are going to make a tail, but these cells back here are going to make a head. They were near neighbors before you cut them apart. Why are they doing different things? How do they know? So it turns out we can visualize a bioelectrical pattern memory in this fragment that says one head, one tail. And that's what they built. And so we figured out a way to rewrite that pattern to say, no, actually two heads. And it's a little messy, but it works, and the technology is still being developed. If you do this, at first, nothing happens. The body is still normal, one head, one tail. The molecular biology is normal. The head marker here is expressed in the head, not in the tail. So there's nothing that you would see different about this body. The anatomy is normal. The molecular biology is normal. But what's not normal is the stored internal representation of what I will do if I get injured at a future time. It's actually a counterfactual memory, and we can read those memories out of the medium non-invasively. And if you do cut this animal, then what happens is the cells consult the memory and they say, a proper worm should have two heads, bang, and that's what they build. And this is not AI or Photoshop or anything. These are real animals. And so what you see here is that we're starting to learn how to not only communicate signals, but to actually rewrite the goal states, right? Rewrite the patterns to which the collective intelligence is trying to reduce error. And if we do that, we find out that it is in fact a very appropriately called a memory, because if you take these two-headed animals and you amputate the ectopic secondary head, you amputate the primary head, they will continue to form two-headed animals in the future. There is no genetic change going on here. We haven't touched the genome. There's nothing genetically wrong with these guys. You can sequence the genome all day long. You'll never be the wiser that they actually would make two heads. And it's a lineage that, as far as we can tell, permanently has been changed. Its sensory motor and well, in fact, in general, the body architecture has been permanently changed because we have reset its bioelectric memory. Not anything having to do with the hardware. So the hardware is wild type and intact, but the software has now been rewritten in place, and you can see what these guys are doing. So this is the amazing aspects of this living material. Evolution gives you hardware that is incredibly reprogrammable. It is really that layer of morphogenesis between the genotype and the phenotype is highly intelligent in the sense of problem solving, which actually makes evolution go quite differently than the standard model. But I probably won't have time to talk about all that. What I do want to switch to now for the last part of the talk is to show you the implications of this going forward. Because so far everything that I've shown you, so the extra eyes on the tadpole and these extra planarian heads and all of that, these are all natural. These are all the normal evolutionarily provided kinds of systems that you've seen. But I want to talk about some novel forms, and we'll warm up to that by looking at the effect of a non-human bioengineer. So this little wasp, what it does is it leaves prompts on plant cells to cause the cells to build something like this. Okay, these are called galls, and they're incredible structures. We would have, if not for this, we would have absolutely no idea that the plant cells, which very reliably build flat green leaves of a particular structure, were even capable of this, right? The latent space of possibilities for the morphogenesis, we'd have no idea that this is possible if this thing hadn't discovered over millions of years, and this is what we're going to speed up greatly, as I'll show you momentarily. We would have no idea that they're even capable of it. And just to be clear, they're not made of insect cells, they're made of the plant cells. And it's not made the way that the wasp creates its nest, which is like a 3D printer where it goes along and sort of puts down every particle. No. What it does is it leaves instructions, prompts that then cause the plant cells to change what they do. We can do some of this too. We can take this planarian. So this is a nice triangular-headed Dugisia doratocephala, and we can amputate the head and perturb the electrical decision-making, the memory of what kind of head should I make. And this can make flat heads like a P. felina. It can make round heads like an S. mediterranea. Of course, it can make its own head. There's about 100 to 150 million years of evolutionary distance between these guys and this. Again, no genetic change. We haven't touched the genome. So what you're seeing here, and it's not just the shape of the head, it's also the brain, the shape of the brain and the distribution of stem cells is just like these other species. What we're seeing here is that perfectly wild-type hardware is happy to visit other attractors in that anatomical space, right? So in anatomical morphospace, you have other attractors that are normally occupied by other species, but this hardware can visit it too. And so the navigation of that space, there's a lot of decision making that goes on about where should we go, but you can impact that if you can communicate with the material. And so now we want to ask the following question. Okay, evolution has given us certain attractors in that space. What else? What else exists in that space that we may not know about? And I'm going to show you a couple model systems for that, but I just want to make this larger point. And if anybody's interested in this, you can come here. We have an asynchronous symposium going on all fall about the nature of this Platonic latent space of form and behavior. I think that everything that I'm about to show you, so the Xenobots, the Anthrobots, the Mombot that we've built, all of these things are vehicles. They are a kind of, among other things, they are our periscope to peer into this Platonic space of possibility to understand what else, what other patterns can come through the interfaces that we build. And these interfaces are cells, biobots, chimeras, AIs, embryos, everything that we build, I think, is a kind of interface to patterns from this latent space. And some of those patterns are the kinds of things mathematicians have been studying for thousands of years. So, sort of low agency things that can be formalized easily by mathematics, and which you can think of math as the behavior science of those kinds of forms, but also much more complex active forms that we would recognize as kinds of minds. And I think that understanding what exists in this latent space and how it relates to the physical pointers that we make, the architectures that we build, is really the kind of the future of all of these fields. And so I want to show you a couple of examples of this. Let's explore that space and see what can happen. And so the first thing we'll look at is Xenobots. I'm just gonna, sorry, I'm gonna pause this here. Yeah. So what we've done here is we've taken some cells from an early frog embryo. So these cells that are going to be the epithelial cells, and we take them off and we dissociate them here. And they could have done many things. They could have died. They could have crawled off and gotten away from each other. They could have formed a nice flat two-dimensional layer like cell culture. But instead, what they do is they assemble into this kind of creature. And I'm just going to show you close up what that looks like. So this is a close up. Each one of these circles is a single cell. Okay, so all of these are cells, and it's just fun that this looks like a little horse. They don't all look like that. But what you'll see is that these groups of cells have very stereotypic kinds of collective movements. And so here it sort of wanders over here, and it has some movements, and it's sort of some exploratory signaling that it's doing here. You have a calcium flash. The flashes are calcium signaling, which basically indicates computations that cells are doing. And so these amazing properties, and so they come together, and what they form is what we call a Xenobot. So it has cilia on the outside, so it swims. We don't have to pace it the way that muscle-actuated biobots have to be paced. It is completely self-assembling. It is completely self-motile. It can go in circles. It can patrol back and forth. You can make it into weird shapes like this donut bot that Doug made. They have collective behaviors. They have individual behaviors. Here's one traversing a maze. So it goes here. It's going to take this corner without bumping into the opposite wall. Okay, so there's a corner here, it takes the corner. Then at this point, it spontaneously turns around and goes back where it came from. We have no idea why it decided to do that. One of the most amazing properties that they have is kinematic self-replication. So if you provide them with loose materials, so these are these little white things are more epithelial cells that we've sprinkled into their environment. They kind of do von Neumann's dream of a machine that goes around and makes copies of itself from stuff it finds in the environment. So what they do is they knock the cells into little piles. So they pull them into little piles, and then they polish the little piles. And because they're dealing with an agential material themselves, those little piles mature to become the next generation of Xenobots. And guess what they do? They do the exact same thing, and they make the next generation and the next generation. Okay, so this is a kind of replication cycle for this novel system. Now, notice something interesting. There's never been any Xenobots. There's never been any selection to be a good Xenobot. We don't think any other creature on Earth reproduces with kinematic self-replication. So it's very interesting to think about what the Xenopus genome learned in its time on Earth. Well, we know it learned to do this, so stages of frog development and then some tadpoles, but it also apparently learned to do this. So it can make Xenobots, and this is an 83-day-old Xenobot. It's turning into something. I have no idea what it's turning into. They have distinct behaviors here. We can say when the computational cost for all of this was paid. It was paid during the eons that the embodied genome was bashing against the environment and selection and all of that. When was the computational cost to design all of this paid? This has never existed, and there's no straightforward story of selection for these properties. And so there's a few ways that people try to go with this. Some people will say, well, it's emergent. And you say, okay, what does that mean? And they say, well, it just means that at the same time that you were doing this, you also learned to do this. But first of all, that doesn't answer the question of when the computational cost was actually paid. And second, it kind of undermines the whole point of evolution, which was supposed to explain the properties of a living thing with very tight specificity around the environment that gave rise to it, right? That was supposed to be the whole point. It's the history of environmental forces that got you to where you need to be. That is not a story that works here. So I think we need to go beyond just saying that it's emergent and really understand that latent space of possibilities of why specific interfaces are pulling down specific forms of anatomy, of behavior. There are hundreds of new genes being expressed differently in these guys, about 600 new genes. So transcriptional behaviors, where are those coming from? And can we learn to predict and to manage them? Now, you might think that this is some kind of weird amphibian thing. Frogs are plastic and embryos are plastic. And so maybe this is some kind of very specific frog thing and so on. Well, I'll ask this question. What would your cells do if we liberated them? Well, this looks like something we might have gotten off of the bottom of a pond somewhere. It looks like a primitive organism. If I asked you what the genome looked like, you might guess that it has a primitive kind of organism's genome. If you were to sequence it, you would find 100% Homo sapiens genome. These are Anthrobots. They self-assemble from tracheal epithelial cells, so from lung epithelium, taken from adult human patients. Not embryos, no, nothing embryonic going on here. Adult, in fact, usually elderly patients give epithelial biopsies. We buy the cells, and we have a protocol in which they self-assemble into these Anthrobots. We do very little. There are no scaffolds. There are no nanomaterials. Much like with the Xenobots, there is no synthetic biology. We don't touch the genome. There are no weird drugs in here. They're living in basically the same environment, except that they've been liberated from the rest of the instructive influence of cells. And as I've shown you before with that example of the tail grafted onto the side of that amphibian, and in general, in bodies, the name of the game is cells hacking other cells to do things, to align them towards goals that they don't know anything about. Right? And so what we've done is liberated these guys from those influences in the body, and this is what you get. You get a different type of creature that's never existed on Earth before. It doesn't look anything like a stage of human development. They have weird properties. For example, one of the things that they do is if you plate a dish of human neurons, so the red stuff here is human neurons, and then you take a scalpel and you put a big scratch through the middle, and you throw some Anthrobots into that culture, they drive down the scratch, they find a spot to settle in. In fact, they combine into this, we call this a superbot cluster. And then what they start to do is this: they start to knit the neurons across the gap. Right? If you lift them up, this is what you see 4 days later. This is where the Anthrobots were working. They try to heal the damage. And that's just the first thing we notice. No doubt they do many other things. But who would have thought that your tracheal cells, which sit there quietly for decades in your airway, have the ability to move around on their own as a coherent little creature and heal your neural wounds, right? It's an amazing, I find it very sort of positive and optimistic that the first thing we discovered from this novel being is a very benevolent kind of event. It's actually helping to heal these neurons. They're an amazing creature. They have 9,000 differentially expressed genes compared to what they were doing in the body, almost half the genome, right? So their transcriptional profile, their behavioral profile, their anatomy, completely different from what they were doing before. Where does it come from, right? So we need to understand this. And they have 4 different behaviors. You can draw a little ethogram of the probability of shifts between the behaviors. 4, not 12, not 1, but specifically 4. Where did this come from? It wasn't selection. Okay, there wasn't selection for any of this stuff. So I think it's really important that we now understand that because of the interoperability of life, because living things are constantly solving problems, they make few assumptions about their own structure, about the outside world. They have to make the best of every situation. Life is incredibly interoperable. Almost any combination of evolved material, engineered material, and software is a viable embodied mind that will be the benefit of ingressing patterns from this latent space. And we will have, as we already do, cyborgs and hybrids and chimeras and synthetic beings of every kind. All of Darwin's endless forms most beautiful are like a tiny little corner of this space of possible beings. And so we need to really ramp up our game in being able to recognize and relate to all of these different kinds of novel embodied minds with which we will increasingly share our world. And so for the last few minutes, what I want to show you is our attempt to develop tools for this. And really, we take this continuum very seriously in that we're developing tools, but we're also developing colleagues in a way. And I think we're increasingly going to see this shift across the spectrum. What we would like to do is to develop a discovery engine that can do the following cycle. It should be able to make hypotheses about what stimuli to be given to cells to get them to build a particular kind of multicellular organism, right? So this is that anatomical compiler idea I spoke about. And then what it should do is it should actually test those hypotheses. It should build a living system with those stimuli given. It should observe what happens and revise its own internal model of the morphogenetic code and the communication with that system. In other words, we want a robot scientist in the lab, an AI-powered embodied robot scientist, which has a mind that thinks about hypotheses of how to talk to cells, but then also a body that enables it to actually physically communicate with those cells and thus refine its view of the outside world, right? So again, you're seeing this. What I started out by mentioning is this symmetry of active inference, of making hypotheses, of being surprised when expectations are not met, revising your priors, and engaging with the physical world to solve problems, right? This is science, but also the exercise of being of any agent. And so we want this discovery engine for communication and collaboration with agential materials. And so we've started to build one. And the specific stimuli, we're going to start with, there are many things that are coming, but we're going to start with stimuli in 4 categories: chemical signals, electrical stimulation, vibrational, so biomechanical, and temperature gradients. We're going to start with those. And with our partners at Boston Engineering, and this is a project that we're doing with a Bongard lab at University of Vermont. So Josh and I and Doug Blackiston, who started out in my group and now has his own lab, we're all collaborating to build this system to try to understand what it takes to build really an autonomous communication interface to the living material of biology. And so this is what the individual plates look like, in which different hypotheses are to be tested, right? So these are not artists' renditions. This is literally what's being built in our lab. Here's what the thing looks like. Josh and I are both Futurama fans, so we tend to call this thing Mombot. This is it. The first iteration. This is version 1.0 standing in my lab. So you can see here what it physically looks like. There's a little close up, and so what I want to talk about is just a little bit is the internal structure, because it is in fact an embodied AI scientist working in the field of synthetic morphology. It has a mind, and much like ours, that mind contains multiple cognitive submodules which have their own distinct individual goals, which are all communicating with each other and proposing hypotheses and in a way competing for attention of the large scale brain, because there's only so many experiments that are actually going to be done, right? So this is highly parallelized and scalable. And then it has a body. The body are these elements that I just showed you. They are again parallel and nested. So many of these things are activated at different scales. So it is this kind of multi-scale architecture. And one of the things that it does, and so here that I'm running out of time, so I'm not going to go through all of the architecture, but there will be a paper. Doug and Cam are writing a paper on this soon. That what it's actually doing is infotaxis. It's searching for more information by doing experiments. It tries to do the most informative experiment to give it the best way to revise its priors and to learn about the world that it lives in. That world is the world of morphogenesis. It's the world of behavior shaping cues that have particular results in anatomy. And so I just want to point out something interesting, that this, again, to go back to this, going back to this idea of doing experiments and revising your revising your picture of the world. This system, it does that at multiple scales. So at one scale, it has hypotheses about the biobots, the Xenobots in particular, that it's building, and it revises those. But even the subsystems within that robot have their own proprioception loop. So for example, this thing, and unfortunately there's a video that should be playing here that seems to not be playing, but there's a pipette that comes down, grabs these plastic, the disposable plastic pipette ends, and so that it can transport Xenobots from one well to another, or chemicals in and out of the solution. It has sensing through that tip, right? It's actually able to know whether that tip is touching liquid or not, right? So there's a sensing mechanism, and there's an internal loop that will adjust its own behavior depending on what it touches. So just like we have large scale goals, and then we have sort of smaller loops within us that can guide our behavior when you reach out and balance balance objects and so on. There are nested proprioception loops inside of this large scale operation. So we're incredibly excited about this, because not only is it going to help us understand how to make and how to communicate with Xenobot materials, it is also, I think, a model system for creating a kind of exploratory AI system. So here are some next steps. First of all, of course, we're going to be using it to develop new kinds of Xenobots, and that's kind of the immediate next agenda. One thing you might think about is adding a kind of curiosity, a metacognitive loop, where it should find new problems to solve, not just solutions. So what it's doing right now is we, as the users, ask for bots that have specific properties, and then it tries to figure out, how would I make a bot to do that, right? So bespoke synthetic living machines, it tries to solve our problems. In the future, there should be components added for creative exploration, where it can pose its own problems and actually say, you know, what would be interesting is a bot that does this and this, right? And finally, what I think would be really interesting is to enable this system to actually communicate with the bots and find out what they want. And what I mean by saying what bots want is the following. You can easily imagine a system where, let's say you have some kind of cell culture system, and there's instrumentation that monitors metabolic stress and other behaviors. And when the cells could learn to activate specific behaviors to get nutrients, to get the machine to put more nutrients into the system, right? They already have this for plants. They have a thing that kind of waters plants when they ask for it. And so you can sort of learn to run parts of your world to help you achieve goals. From that kind of instrumental learning, all the way to a very sophisticated hybrid system that contains us as scientists, it contains the Mombot, it contains Xenobot material, in which really the behavior of the bots control and give cues to the Mombot to get it to do new experiments to change the way the bots are made, and back and forth through that cycle to make an agential material that actively collaborates with its environment. And it can do that because the environment is high agency too. The Xenobot's environment contains not just the passive water in the dishes, but it contains Mombot, and it contains us as engineers, right? So the material has the opportunity to collaborate with the environment to shape its embodied minds and the future of its evolution. And I think the future impacts of this is going to be along three basic tracks. First, just the obvious thing is useful synthetic living machines. So in the body repair, environmental cleanup, exploration, maybe space, those kinds of things. Then the second thing, the more long-term thing is this, robot science to help crack this morphogenetic code. How do we communicate with groups of cells to regenerate after injury, to repair birth defects, to normalize cancer cells and all of that? Really to try to help us develop this anatomical compiler, right? So this is I think the future of science, is embodied AI to help us crack these kinds of problems. But also very important is something quite different, which is the use of the system to communicate with unconventional minds to help us. So this is the diverse intelligence research program to help us recognize very alien kinds of minds that I think exist all around us, to actually join with them, to be parts of collaborative, aligned, cooperative systems, and actually be part of it. And one way to look at this is not just as the automation of discovery, not just as an anatomical compiler, but the Mombot platform as a communication node, as a sort of a bow tie node in between human scientists and the cellular collective intelligence. Right? One way to look at this is as a translator interface, but it's not a passive translator interface. It itself is an active intelligence. And this idea of having AIs and in general nested cognition that becomes interfaces between other cognitive systems, I think is very powerful. And it particularly is critical because of the need to develop an ethical synthbiosis. We are going to have to, I think, if we move forward as a mature society, we are going to have to learn to relate to these radically different kinds of minds. So I'm gonna wrap up here and just remind us of the main points that I tried to transmit. Morgan's canon, which is basically this idea that you should always skew low on assays of intelligence for whatever you're looking at. You should assume it's a dumb mechanical system. I think it's an off-ramp for science and engineering. I think it's really limiting in biology, in bioengineering, in biomedicine. I think it's a big mistake to do that, as opposed to developing tools that allow you to guess correctly, not just skew low, but guess correctly. I think there's a really profound symmetry between the cognition of agents and the process of science. So the process of discovery of the self, of the world, and how you enlarge your perspective upon new minds and to actually collaborate with those new minds to enlarge your perspective. And I think now we can develop tools to rigorously define what actually exists on that spectrum of persuadability that I showed you, on that cognitive spectrum, to communicate to them via novel interfaces, be they bioelectric, biomechanical, and things we haven't even thought of yet, and then to develop new ways to relate to them, because I think that AI, ALIFE, and the understanding of agential materials is really the future. So I'm going to end here by doing the most important part, which is to thank all the people who did the work. And so here are the postdocs and grad students and everybody else who did the work. In particular, for that last part of today, I want to really thank my close collaborator Josh and his whole lab. Like this is his team, all the people that have worked on the cognitive side of Mombot, Boston Engineering, who are the company that helped us build all of these things, and their team. Doug, of course, in his new lab. Lots of funders in the past that have supported various efforts, but Krell in particular that has really funded us to make this quite expensive engineering effort. And shout out and disclosures of three companies that I want to mention. So Fauna Systems is our partner for taking forward the actual Xenobot technology. So whatever it is that Mombot discovers, Fauna Systems is our partner for that. Astonishing Labs, which are supporting us for the study of different kinds of cellular intelligence and bio-inspired AI and many things like that, and Softmax as well. And you heard from Adam Goldstein, this business of aligning parts, and in particular alignment between different scales of cognition, and using a bio-inspired cognition first approach to alignment between artificial systems and humans, I think is incredibly important for us going forward. And a huge shout out to Marty Schwalm, the amazing engineer who made the robot through which I'm giving this talk today. Thanks so much, Marty.